Hi team, today we're going to be talking about gluconeogenesis. So that's going to focus on these bottom 4 or 5 learning objectives relating glucose metabolism to insulin and glucagon signaling, comparing and contrasting glycolysis and gluconeogenesis. We're going to explain how gluconeogenesis is powered in the cell and how gluconeogenesis gets around these irreversible glycolysis reactions. And we're going to also mention briefly the Cori cycle and what it does for the body. Remember, when we're thinking about glucose metabolism, it has a lot to do with regulating glucose levels in the blood. So our blood is at some normal level of glucose, and after we eat a meal, we're going to have an increased level of sugar in the blood. We'll be hyperglycemic, that is, we'll have more than the normal level of glucose. That will stimulate the pancreas to release insulin. When insulin is perceived by cells in our body, glucose is uptaken by the cells and used to produce ATP and energy inside our cells. And in the liver, glucose can also be stored as glycogen. So glycogenesis is stimulated in the liver. In both of these ways, cells in the rest of our body, like muscle cells or neurons, as well as cells in the liver, taking up glucose, that's going to return our plasma glucose levels to normal. We can also think about what happens when we get low on blood sugar, when it's been a long time since we've eaten and we have hypoglycemia or low blood sugar. At that time, our pancreatic cells will release the hormone glucagon, and glucagon stimulates gluconeogenesis in the liver. Glycogenolysis is also stimulated in the liver, that is, breakdown of glycogen into glucose. But today we're going to focus on gluconeogenesis, that is, making glucose molecules from other biomolecules that are present in our cells. In both of these ways, glycogenolysis and gluconeogenesis, the glucose levels in our blood will rise and bring our body back to a normal level of glucose. When our pancreas senses a low blood glucose concentration, the hormone glucagon is released, and glucagon binds to a G protein coupled receptor. And that G protein coupled receptor will activate adenylyl cyclase. Adenylyl cyclase is an enzyme that converts ATP into the secondary messenger cyclic AMP. And cyclic AMP has a lot of roles inside the cell, one of which is to activate a regulatory protein that helps a pyruvate transporter move pyruvate into the mitochondria. And pyruvate inside the mitochondria ultimately will lead to the slowing of the citric acid cycle. Citric acid cycle. And the upregulation of pyruvate carboxylase, which is the first step in gluconeogenesis. So we're going to talk about this step of gluconeogenesis today. We've already discussed the pathway shown in red, that is glycolysis, where we start with glucose and break it down into 2 3-carbon molecules of pyruvate. And in this glycolysis breakdown process, we use the enzyme hexokinase, phosphofructokinase, and pyruvate kinase. All 3 of these are highly regulated steps in glycolysis. Ultimately, pyruvate can go on to do lactic acid fermentation, or it can be part of the citric acid cycle. Both lactic acid fermentation and the citric acid cycle are strategies of getting ATP energy for the cell. Sometimes, however, we need to build glucose. And in order to do that, we can, in essence, run glycolysis backwards in a process called gluconeogenesis. You can see that lactate can come back into this cycle to produce pyruvate, and then we'll run glycolysis backwards. You can also see that the amino acid glutamate can enter into the citric acid cycle, ultimately producing oxaloacetic acid, which can then be brought back up into gluconeogenesis, or the backwards citric acid cycle. Last but not least, glycerol from fatty acid breakdown can also join into gluconeogenesis. So all three of these other biomolecules can be used to help build glucose when it's needed by the cell. Of course, gluconeogenesis is more than just the backwards glycolysis. And we'll talk about some of the strategies that your cell uses to get around these irreversible reactions in glycolysis when we want to go back up the chain toward glucose. First, let's look at a summary. In gluconeogenesis, we're starting with 2 3-carbon pyruvate molecules and combining them to create a 6-carbon glucose sugar. Because we're going to be building a carbon-carbon bond, this is going to be a high-energy process. We're going to require ATP, or energy input, into the process. And we're going to be producing pyruvate and ultimately glucose from non-carbohydrate molecules. And this includes glycerol, glutamate, and lactate. And we talked about where those different non-carbohydrate molecules can enter the pathway on the previous slide. One of the stumbling blocks of gluconeogenesis, though, is that we have to bypass the irreversible glycolysis reactions that are catalyzed by hexokinase, phosphofructokinase, and pyruvate kinase. The other enzymes in glycolysis are used in gluconeogenesis. They're just run in the opposite direction given the different concentrations of substrates within the cell. Overall, the cost of gluconeogenesis is 4 ATPs and 2 GTPs and 2 NADHs. We've got 6 high-energy phosphate molecules here, so this is actually quite a spendy process. If gluconeogenesis costs 6 phosphate molecules, is this an endergonic or an exergonic reaction? Well, net, this is going to be an endergonic reaction because we are building up. That is, it's an anabolic reaction, and those are always endergonic. But it's because we're coupling these endergonic reactions to the highly exergonic hydrolysis of these phosphate molecules that cells are able to perform this process in a thermodynamically viable way. Let's compare for a second glycolysis and gluconeogenesis. In glycolysis, we're using glucose to produce 2 molecules of pyruvate. 2 ATPs are invested, and in total, we get 4 ATPs out. That's a net of 2 ATPs. And we're going to use these irreversible hexokinase, phosphofructokinase, and pyruvate kinase enzymes. Altogether, the delta G of glycolysis is minus 90 kilojoules per mole. So this is a very exergonic reaction. We're breaking bonds. This is catabolic. In contrast, gluconeogenesis starts from 2 pyruvate molecules to build a 6-carbon glucose molecule. we're investing a lot of energy, 4 ATPs and 2 GTPs. And rather than using these irreversible glycolysis enzymes, we're going to use pyruvate carboxylase, PEP carboxykinase, and fructose 1,6-bisphosphatase. It's because we're using these high energy molecules that we can run gluconeogenesis as an exergonic reaction. So this is going to be net exergonic thanks to ATP and GTP hydrolysis. Without ATP and GTP hydrolysis, this would be otherwise an endergonic reaction. Because we're building bonds, it's anabolic. You can see, though, inside the cell, the delta G of gluconeogenesis is a thermodynamically favorable reaction because we're breaking these bonds of ATP and GTP. We're using them as the source to power our gluconeogenesis. So as we start with gluconeogenesis, we're going to focus on beginning with pyruvate. And this diagram you see, first we move from pyruvate to oxaloacetic acid. And then oxaloacetic acid is converted into PEP, phosphoenol pyruvate. And in order to do that, we need two enzymes, pyruvate carboxylase and PEP carboxykinase. And these enzymes are used to sort of take the place of pyruvate kinase, which is unidirectional. It only runs during glycolysis. Using the standard enzymes of glycolysis, we can go backwards up the pathway to fructose 1,6-bisphosphate. But in gluconeogenesis, we can't use phosphofructokinase. We have to use fructose 1,6-bisphosphatase. We can use the standard enzymes of glycolysis to get us back up to glucose 6-phosphate. Glucose 6-phosphatase can go on to be a part of creating glycogen. But if we need to go back to pure glucose, we need to use glucose 6-phosphatase. Because again, in gluconeogenesis, hexokinase is not available to us. So let's talk about some of these modified steps in gluconeogenesis that aren't present during glycolysis. First, how do we bypass pyruvate kinase? In order to do this, we actually have to use multiple enzymes. We start with pyruvate carboxylase in the mitochondria, and you can see here I've written PC. Pyruvate carboxylase adds a CO2 group to pyruvate to produce oxaloacetate. Oxaloacetate can be readily converted into malate, and malate is easily brought into the cytoplasm of the cell and then easily reversed back into oxaloacetate. The enzyme pyruvate carboxylase in the mitochondria is really interesting because it uses biotin as a cofactor for enzyme activity. It carries an activative CO2, and this is what's added to pyruvate to turn it into oxaloacetic acid. Another fun thing about pyruvate carboxylase is that it performs a double displacement or ping-pong reaction. And we haven't talked about reaction mechanisms too much in this class, but since this is a fun example, I thought we should talk about it. Let's look first at the top here. We've got pyruvate as well as ATP and CO2 as our substrates. Our biotin cofactor, as well as a magnesium ion, are important for pyruvate carboxylase to work. Ultimately, we end up with oxaloacetate, as well as ADP and PI leaving. The way this reaction actually takes place inside the enzyme, however, has ATP joining CO2 in binding to the enzyme. So you can see first CO2 and ATP bind to the enzyme biotin complex. The first reaction occurs so that we have biotin now binding this activated CO2 and ADP and PI being hydrolyzed. We've used the energy of ATP hydrolysis to build this activated biotin carrier. Next, pyruvate joins the enzyme, and pyruvate can interact with the activated CO2 on biotin to form oxaloacetic acid, which then leaves the enzyme. So we've got some products leaving before pyruvate ever even joins the reaction, and ultimately pyruvate leaves as oxaloacetic acid. Pyruvate carboxylase. We're adding a carboxy group to pyruvate. All right, we've just talked about pyruvate carboxylase in the mitochondria. Once we get this oxaloacetic acid made from pyruvate carboxylase into the cytoplasm, we need to turn oxaloacetate into PEP, or phosphoenol pyruvate. In order to do that, we use phosphoenol pyruvate carboxykinase. We need to add a phosphate group to oxaloacetate in order to make this reaction work. And here we're using actually GTP hydrolysis to add this phosphate group and produce PEP. Additionally, you can see we're releasing CO2 in this step. So we've used the carboxylation or decarboxylation to create this high energy molecule. Now, if we were going to try and reverse pyruvate kinase from glycolysis, the delta G of the reaction is over 30 kilojoules per mole. That's just not going to happen. However, by using pyruvate carboxylase and then PEP carboxykinase, the delta G of the reaction is only 0.8 kilojoules per mole. And we're using ATP and GTP as well as this decarboxylation reaction in order to lower the delta G of the reaction to get it to a thermodynamic level where it will actually occur inside the cell. Okay, we've successfully bypassed pyruvate kinase. Let's talk about the next enzyme in the pathway. We can use the standard enzymes of glycolysis to get from PEP to fructose 1,6-bisphosphate. And now we would normally use PFK to get back to fructose 6-phosphate, but PFK again is irreversible in glycolysis. Instead, we'll use fructose 1,6-bisphosphatase. So phosphatase is the opposite of a kinase. We're starting with 2 phosphate groups on fructose and removing one with the help of bisphosphatase. So here's our removed phosphate group. Look at this beautiful allosteric enzyme. You can see it's got 4 identical subunits. It's a tetramer, and that even just structurally suggests that this is an allosteric enzyme. And it is allosterically regulated in a way that's opposite of phosphofructokinase regulation. That is, where ATP inhibits phosphofructokinase, ATP will activate fructose 1,6-bisphosphatase, and AMP will activate phosphofructokinase, but AMP will inhibit fructose 1,6-bisphosphatase. From the citric acid cycle, PFK is inhibited by citrate, but fructose 1,6-bisphosphatase is activated. This makes sense. If we have a lot of the downstream products of PFK, like ATP or citrate, this can signal to the cell that we don't need to run glycolysis quite as quickly. We can slow things down. Whereas if we have high levels of ATP and citrate in the cell, it will signal maybe we should produce glucose and then store glucose as glycogen by running gluconeogenesis. It's this reciprocal regulation of PFK and fructose 1,6-bisphosphatase that allows our cells to use the same enzymes for glycolysis and gluconeogenesis, but never run the same pathway in a single cell. So if fructose 1,6-bisphosphatase is running, we know that PFK is going to be inhibited. So in a cell where this is true, we'll be doing gluconeogenesis and not glycolysis. However, under cellular conditions where PFK is activated, those same conditions will inhibit fructose 1,6-bisphosphatase, and so we'll do glycolysis and not gluconeogenesis. Reciprocal regulation. To get all the way back to glucose in the gluconeogenesis pathway, we need to bypass hexokinase. This is often not a step that's needed because G6P or glucose 6-phosphate can be used elsewhere in the cell. That is, it can be stored as glycogen or it can enter the pentose phosphate pathway to create nucleic acids. However, if we really need to be regulating blood glucose levels, we do have to produce unphosphorylated glucose for it to be in the blood. And the liver can produce glucose from G6P in the endoplasmic reticulum. So glucose 6-phosphate enters the endoplasmic reticulum. It's converted by glucose 6-phosphatase into glucose plus inorganic phosphate, and then this glucose can be released ultimately into the bloodstream. Okay, we've talked through starting from pyruvate and with a detour through oxaloacetate, we've returned all the way to glucose using gluconeogenesis, and we've used 4 new enzymes to get around the irreversible steps in glycolysis. We've used pyruvate carboxylase, pep carboxykinase, fructose 1,6-bisphosphatase, and glucose 6-phosphatase. Similar to those unique enzymes in glycolysis, these 4 important enzymes are often heavily regulated by the cell in order to determine whether gluconeogenesis is moving forward quickly or slowly. Again, in summary, in red is shown glycolysis. And I'm only showing you here the regulated allosteric steps of this pathway. So glucose to glucose 6-phosphate is catalyzed by hexokinase during glycolysis. We get to fructose 6-phosphate, and that is catalyzed by phosphofructokinase into fructose 1,6-bisphosphate. Multiple steps in the pathway pass by, and ultimately phosphoenol pyruvate is converted into pyruvate by pyruvate kinase. These are our 3 most heavily regulated enzymes, all allosterically regulated in glycolysis. Now, running in the opposite direction is gluconeogenesis. We start with pyruvate at the bottom here, and we use two enzymes to bypass pyruvate kinase, going through oxaloacetate. From PEP, it takes several steps. Using the same enzymes as are used in glycolysis, we can produce fructose 1,6-bisphosphate. With fructose 1,6-bisphosphatase, we can then produce fructose 6-phosphate, which can be converted back to glucose 6-phosphate, and ultimately glucose 6-phosphatase allows us to rebuild glucose, which can go off into the bloodstream as needed. As discussed, those key regulatory steps in glycolysis are also the regulatory steps in gluconeogenesis. And because these two pathways are reciprocally regulated, that means that only glycolysis or gluconeogenesis can run in a given cell at one time. Glucose 6-phosphatase inactivates hexokinase, but it can activate glucose 6-phosphatase. We've got allosteric regulation of phosphofructokinase. You can see that some of the same things that activate fructose 1,6-bisphosphatase are inactivating phosphofructokinase and vice versa. At the bottom here, our PEP pyruvate enzymes aren't reciprocally regulated, but you can see that ATP is inhibitory to pyruvate kinase. So if there's lots of energy inside the cell, pyruvate kinase is inhibited. Whereas ADP, the breakdown product of ATP, is what's inhibitory to gluconeogenesis. So if we have some of this building up saying we've used up ATP, that's going to inhibit the gluconeogenesis side of the pathway. So they're approximately reciprocal in their regulation. This interregulation of gluconeogenesis and glycolysis is called the Cori cycle. In muscle cells, for example, glycolysis is being used to produce ATP. After pyruvate is produced, it can go on to citric acid cycle and electron transport chain to produce ATP. In the absence of oxygen, though, pyruvate is fermented into lactate. And this lactate can build up in the muscles, and in the muscles, it's a dead-end product. Instead, we need to ship lactate through the blood back to the liver. And in the liver, the enzyme LDH is available to turn lactate back into pyruvate and ultimately use gluconeogenesis to build pyruvate back into glucose, which can again be made available to cells in the body for use in glycolysis. The Cori cycle refers to this shared responsibility, this symbiotic pathway between the muscle cells that are producing lactate and the liver cells which are utilizing lactate to produce glucose. It's important to note here that glycolysis by itself produces about 2 ATPs, whereas gluconeogenesis, as we discussed in this video, costs 4 ATPs and 2 GTPs. So it's quite energy intensive to rebuild glucose from lactate, but it allows the body to regulate its blood glucose levels and eliminate this lactate waste product. Carl and Gertie Cori were from Austro-Hungary and emigrated to the U.S., and they were awarded the Nobel Prize in Physiology and Medicine in 1947 for their work on the catalytic conversion of glycogen. Now, the Cori cycle is named after them, and they described it, but their Nobel Prize was not for the Cori cycle. Instead, it was for the larger work with carbohydrate metabolism and glycogen. And we will go on to talk more about glycogen metabolism, its synthesis, and its breakdown in coming chapters. Fun fact, Gertie Cori was the first woman awarded the Nobel Prize in physiology or medicine. All right, with questions about gluconeogenesis, please email me or bring them to class. Thank you.